Thank you very much. And I'm here to follow nicely on what Anthony's described, but perhaps the same message about the importance of having medical input and pathology in the short term. It'll change in the long term. But not all of you come from a pathology background, not all of you come from a genetics background. So what I'd firstly like to do is just put some of the terms in context, go through and describe where genetics ends and where genomics begins. The two terms are not interchangeable. Then I'll run through and show where we are with omics, mainly genomics, as of Australia 2017, where we're likely to go, and then I'll finish by showing you some of the big data challenges that we have. If I get through it all in half an hour, great. If I'm really, I've dosed myself on coffee just before coming in to make sure I maintain the pace, because this morning while sitting at the table, made a genetic diagnosis using genomic techniques. It can all be done over even our limited bandwidth here. And I'd like to show you that patient, so you can actually see these things applied in real time to a real live patient, if we have time. So keep me moving, keep me moving. So what is genetics? What is genomics? Well, genetics hasn't changed. It's still the study of heredity and inherited disease. And it tends to be, because of the technology, one gene or one phenotype or one family at a time. And genomics gives us the ability to study all the genes in the genome at once. And I'll show you examples of how they apply later. There are different types of genetic disorders. The ones that Anthony was starting with the pancreatic cancers, were the somatic ones, where you generally acquire the variant or the mutation during life. And the ones that I tend to be more interested in are the germline ones, which you're born with and you carry, and they may have a predisposition for cancer during life, but generally you get them from your parents, you pass them through to your children, and as I'll show you, you can also acquire a couple that then become part of your germline to go on. There's also a third type, epigenetics, which I won't go into, which is where you acquire through life modifications to your DNA that you'll pass on to your children and your grandchildren, but which will then disappear from your lineage. So they're only present for up to 3 generations. And the testing also differs. In genetics, we tend to test either individuals or perhaps families, while genomics, you do individual, families, cells, tissues, tumours, microbes, and it's complex and it's big data, it's huge data, and I'll show you later some of them. And the key thing about the omic technologies is that you're not really fussed whether you're doing 1 or 10 or 100 or 20,000 genes, it's all the same. You just start at one and you sequence the whole lot and you let the computer sort it out. What can you do with omic technologies? Well, diagnosis. We're all in the medical field. So diagnosis, you can diagnose someone who's got a condition, a complaint or a presentation, I'll show you, and you can start applying to personalised precision medicine, as Anthony was hinting at, but there are a number of conditions, perhaps not so much in the somatic cell area, but in pharmacogenomics, where you can match the patient with their appropriate medicine. Most of you after lunch today and probably after dinner this evening will have had more, and after the hors d'oeuvres last night, the canapes last night, will have more than your fair share of oils and fats and all the foods that your doctor tells you not to eat. And some of you are probably on statins. Well, there are 3 or 4 classes of statins, and some of you are going to get a myopathy from it. And we can predict by measuring your genetic markers which of you are going to get the myopathy and which of you are not. So you've got the opportunity to avoid the adverse drug reaction before we even start, just by doing the genetic testing. As well as genetics, you can go to screening, which is where my passion has been. Why wait till someone gets ill? You can go out and look at either for an individual or a family or even an entire population and look for particular genetic markers that will determine the predisposition to various conditions. You can screen healthy populations. You can screen populations that come from geographic regions where you know there's going to be an increased or decreased ethnic risk. And there's a direct-to-consumer, which Anthony covered very nicely. And within the diagnostic area, again, you can apply in different areas. You can do it one person at a time, proband, but you can also do the child and the mum and the dad. And the power of doing all 3 at once. You can see which of them were inherited from the parent and which of them are de novo mutations, which started for the first time in that individual. You can do a diagnostic odyssey, which is a patient I hope to show you later on, time permitting, which is what you do when all else fails. Those of you who haven't yet seen the Ask the Doctor series on ABC, 4 weeks ago on ABC, you can still see it on iView, there was a program called Ask the Doctor on Genes, and it showed the story of young Alan, who was a young 8-year-old boy who was going to die, and we identified the gene in him, found an unrelated drug that had never been used in his condition before, and miraculously his condition stopped and he was able to go 2 weeks later to the opening of Star Wars. So you can match the drug to the patient in a diagnostic odyssey where otherwise the diagnosis would never have been made. And cost effectiveness, again I'd agree with Anthony's comments that there are very few conditions as yet where we have clear evidence that genomics is going to be ahead of genetics, is going to be ahead of other pathology. Lots of very promising ones, and the studies are underway, and we'll know within the next 12 or 18 months which traditional pathology test will be retired in favour of going down the genomic pathway first. Treatment, I've mentioned pharmacogenomics, both for adverse drug reactions as well as to match the correct patient to the correct drug so that you don't have any side effects. The targeted treatments, which again Anthony's hinted at, and you can also use systems biology, as happened in this Ask the Doctor program, where the drug they used on the young boy had never been used in that condition before, but we're able to identify the molecular pathway and suggest that a drug in another disease would work in this child, even though it had never been used before. Screening populations, healthy adults, we can predict which of you will be at increased or decreased risk of getting cancer or cardiac disease later in life, or even population screening, where some of the early studies are being looked at to see whether genomics can be used in the newborn, and if so, whether it's better, the same as, or not as good as our current newborn screening programs. So much for where you can use the genomics. How do you actually use it? Well, it's currently used mainly within pathology, that's where it started, but it's going to spread through so many other areas of clinical medicine. Within pathology, we mean by this that it's an evidence-based discipline. We're searching for genetic variants, and I'll show you in a moment, there are a dime a dozen, but which of them actually correspond to disease, either disease causation or disease treatment. And for genetic pathology, in its current incarnation, it's basically the process of searching for abnormalities or changes in the DNA and comparing the changes in your DNA against the DNA changes in the DNA in a hypothetical normal or reference or unaffected individual, and then trying to work out which of those changes or variants corresponds to the condition that you have. But the problem with it is that patients don't present with genes, and they don't present with diagnoses, they present with symptoms and signs, or aches and pains or complaints. So the way that medical practice is, you go to see your doctor and you unload your latest list of aches and pains, and the doctor comes up with a working diagnosis, differential diagnosis, and think, well, I'll just do a test to confirm if this is it, or a test to rule it out. And if it fits, then good. But if it doesn't, you go back and you revise the diagnosis and you apply another set of it. It's time, it's tried and true, it's tested over time. The process works, but it really works only when there's a small number of tests that can be applied against a small number of conditions. So in the history of genetics, we've gone through starting with single genes and going up to whole genomes. With single genes, we're doing it one gene at a time. Cystic fibrosis would be the most common approach. Perhaps hemochromatosis. It's cheap, $50, $100, $200. It's simple. Won't be around for much longer. Gene panels, if you know that you're looking for one gene, and this gene often tends to cluster in a group with other related genes, or you're looking for the genes that can cause a condition, why not construct a panel, a group of genetic tests that will always be done for the same person? A test for someone with a neuromuscular disorder, or a test for someone with a respiratory disorder, or a test for someone with a cardiac disorder. Now, they generally have about 20 to 200 genes in the panel. They're expensive to develop, but once developed, they're very cheap to run. Somewhere in the few $100s is a typical price for a panel now. The only problem is, with every gene discovery, your heart lifts. There's a new cause of disease. And then you grimace when you realise your panel has suddenly become out of date. And you're going to have to spend the next 12 or 18 months revalidating it. And then you have to rebuild the panel, knowing that by the time you've revalidated, someone else will have discovered another gene that should have gone in. So the trouble with gene panels, they're locked into a cycle of 12 or 18 month validation, so they're always out of date within a few days from the moment you release them ready for testing. So the next step was, well, why go to all the trouble of validating a panel? Why not look at all the genes? We know there's about 20,000 genes, only 4,000 we have a disease associated with them, so you can either do a whole exome, 20,000, or a clinical exome, the 4,000, and do them all at once. And then don't worry about which panel is current. Just do them all, let the computer sift through and put to one side the ones we don't know the cause. It's complex, feasible. Current cost is probably around $1,500. Diagnostic rate, 20–30%. In fact, one step further, this is probably the most widely used slide in genomics. It shows you the falling costs over time of genetic testing. 1 year ago, the cost was $1,000 actually $1,100. That's for the reagents alone, the chemicals alone. When you consider that the cost of testing is $2,000 for an exome to $4,000 for a genome, that's where the point that Anthony was making, most of your cost is not the chemicals. Most of the cost is the infrastructure and the professional work that goes in trying to sort out what all the information means. But this cost is going to drop further. It'll probably drop to around $100 sometime in the next 12 or 18 months for the reagents only, but the rest of it won't change. It'll probably become even higher because the amount of data will be even more complex. So the whole genome became possible 2 years ago as a result of a very generous donation given to the Garvan by the Kinghorn Foundation. We purchased Australia's first and still the only whole genome sequencing system. We were the second accredited site in the world, and I think one of only either 3 or 4 still at the moment. The whole genome, we sequence everything, but we then bioinformatically discard everything, and we actually bioinformatically do an exome. So a whole genome is actually a whole exome, but done with a different technology, which has a much more uniform coverage, and therefore you don't skip so many of the genes. Because of the way it works, it has about twice the diagnostic yield, about 60%, 58% is in our experience, for selected conditions, and probably about 50% for unselected. And the cost is higher per patient, but the diagnostic yield is higher by the same amount. So again, the health economists are trying to work out whether the sweet spot is exome or genome, and the falling cost of sequencing means that very soon there'll be very few genetic tests done that aren't done by either whole genome or whole exome. Panels are going to be here for a year or two and disappear. Single genes, they'll be there for very restricted. It's all going to go to sequencing. The cost of sequencing the whole genome will become so cheap that no one's going to sequence groups of genes anyway. And the machines are going to shrink and everyone will have one. So the pros and cons. Pros of genomics, it's got the maximum diagnostic power. It finds everything. It's best value per gene, lowest cost per gene. The cons, it's complex. Maximum diagnostic power, finds everything, best value, it's got so much information. And you also sometimes find things you're not looking for. It's the genetic equivalent of having a chest x-ray because you've got a sore shoulder and finding you've got a coin lesion in your lung. You go looking for one gene because you've got one condition, you find you've got another genetic variant, you may or may not want to know about it. So it's very important to address the issue of incidental findings. Before we do any genetic tests, and we only accept referrals from a medical practitioner or a genetic professional, we require that the patient, or the client, has gone through genetic counselling, and the major purpose of genetic counselling is so that we're satisfied that they understand what they're getting into, what we're going to find, what we might find, and that we know what their wishes are. Because the autonomy of the patient, they may say, look, I don't care what you find, just tell me why I've got a sore foot. Well, they might say, while you're at it, tell me if I'm predisposed to a number of... So different patients want different things. Out of interest, I've had my genome sequenced. Where is it? And I did it because I needed to know what the experience was. How am I going to advise people whether or not, or the pros and cons of it, if I didn't know how I felt myself? And I took it as a family discussion before I went ahead. Those of you, when you watch Ask the Doctor, the ABC program, you'll see the same process going on. So with my wife, who also has a laboratory background, we discussed it, and I said, look, I'd really like to go ahead. She said, over my dead body, whatever you find, do not tell me anything. I don't want to know what you've got. I said, okay, fair enough. And then our daughter, who also works in the same field, I asked her opinion. She said, oh, over my dead body, do not withhold anything. I want to know everything. I want a copy of your genome because I share half of that genome with you and it's relevant to me. So even within the one family, you had the complete range of views there. So you need to know what the individual wants, and you're legally and ethically and morally obliged to carry out their wishes. So how does it work? It's a standard pathology test. You start off with a referral, you go through a request form, which can be paper, it can be on the net. You collect currently a blood specimen, which works just as well with sputum or spit or dry blood spot or a whole pile of other tissues. It comes through the pathology couriers into the sequencer, I'll show you that in a moment, into the computing, I'll show you that in a moment, through the pathologist, I'll show you that in a moment, and you get back in this age of modern informatics, the most common form of delivering a genetics result in Australia today is by fax. The information loss is phenomenal. So here we have the state of the art, whole genome, and at the request of the recipient, would you please fax it to me? So we resist as hard as we can. I'll show you we have other solutions available, but that's still how genetics is done. Informed consent, different laboratories, different consent forms. The major purpose is to make sure we know what the patient or the client's wishes are and to make sure we're satisfied they understand the pros and cons of going ahead. And the testing process is pre-test, counselling, just like pathology. You get your DNA, put it through the sequences. I'll show you the machine with the lights that go ping. Bioinformatics, massive data. I'll show you how that works. And the last bit is the pathology, the morphologomics, is that the word? But that's where the professional input goes in, and that's where most of our work and most of our costs go in at the moment. Because the problem is, you've all got big genomes. 3.2 billion nucleotides, haploid, 6.4 billion in a diploid genome, and your DNA polymerase does a great job. It's the most accurate copying enzyme we know, but it still makes a mistake about one position in 1,000. So if you think about it, the person sitting next to you will differ from you by roughly one position in 1,000 in 3.2 billion, 3 million places. And yet the person next to you is probably as healthy as you are, or as healthy as you are, which means that most of those 3 million variants have no medical significance whatsoever. And we have to work out which of them are going to be important, a very small number, and which are unimportant, and just make the person next to you look different. To you, most of them are of no medical significance. So it's a gigantic computational jigsaw. And the process, the first genomes took more than a decade to put together. These days, it still takes us between 2 and 8 weeks of playing with the jigsaw puzzle, although the actual computation time, there are hardware accelerated chips that can do it in about 3 hours, but the rest of it, you've still got the medical input to go afterwards. We start off with the 3 million variants, how you differ from the next person. There are still some areas of the genome we cannot sequence to this day, highly repeated regions, centromeres, highly GC-rich regions, but we get about just under 3 million good calls. Most of them fall between the genes. Only 1% of your genome is coding genes. Of those, many of them are simply swapping one DNA sequence for another DNA that will make the same gene product. Most times it has no medical significance. We then compare to healthy individuals. Most of the differences are found in other perfectly healthy people. They're not the cause of the disease. And from this point onwards, the computer stops and the eyeballs take over. And so we hand curate the last 50, 100, 150, in a difficult case, perhaps the last 1,000. The facility, we're fortunate to have a purpose-built building, an 11-storey building, where 4 of the stories are basically the gene sequencing facility. It looks a little bit like Hogwarts Manor from Harry Potter. It's an architect's paradise. The stairs really do move when you stand on them and they bounce around. The floor numbers do change as you cross from one building to the other, but it really is a very exciting, but you know you're in the future when you're inside the Kinghorn Centre with Genome.One. That happens to be the DNA sequencing floor. We have 2 complete floors of bioinformaticians who drive the machine and another half floor of medical staff, clinical geneticists, genetic counsellors and pathologists. The actual machine, this is, we happen to use the Illumina technology, which is the dominant one in the field. That's 1/10 of an Illumina X10. They come in packets of 10. Special, you can now buy them in packets of 5. They will set you back somewhere between $10 and $20 million. And they're very simple. It's just a simple microscope slide where you put there, a computer screen, computer down the bottom, and that's the world's most expensive bar fridge, which is holding all the reagents. And what you're doing is putting your shared fragmented DNA with all the reagents on one of 8 etched lanes in a glass slide. I'll go back to the previous slide. It goes into this machine, and then through the out of the bar fridge, you suck chemicals and wash that slide with chemicals that will react to the nucleotide A. And you photograph where on the glass slide A's appear, and then you wash it off and wash it with C's and G's and T's and A's and C's and G's and T's and A, and you do that 300 times. And the lasers and the cameras are photographing where the little sparkles of light come, and then through fat data pipes out the bottom of the machine we go down to the server farm in the basement, which is doing nothing other than gathering data. When all 10 machines are running at full speed, it takes up the entire computing power of the building to suck the data out there. And then once we've finished that, we then begin the process of bioinformatic analysis. 10 machines, 9 of them in operation at any one time. 1 is always down for loading, unloading or servicing. The capacity of 9 machines, 8 channels, 3 days running time, at 1 genome every 30 minutes. That's the throughput of and this is the current hot rod of the field, but as of later this year, they're bringing out the next model, which will increase that by a factor of 10, and in theory will drop the price by a factor of 10, although you have to buy the machine. They cost several million each. Okay, so that's the theory, the terminology, that's how it works. Let me show you how you actually put it in practice. It's only going to work if it scales. And the bigger they get, and the faster they get, the cheaper it's going to get, and therefore the greater the number of applications. We started 2 years ago with our first genomes. We've actually hit our capacity this month. Beginning of the month, we have a theoretical capacity of 16,000, but a number of those are quality control samples going through, so 15,000 is the practical throughput. We are operating at 15,000 now, 24/7. The organisation has grown. Currently we've got 60 people looking after the feeding the machines, interpreting them, working out what the results mean. The vast majority of these are research samples, only a minority are going to be actual clinical diagnostic samples. And it's generating massive amounts of data. So the numbers I've rounded them a bit. It takes about 1/4 terabyte per sample. That means we're pushing 150, 200 terabytes per month. We've stored so far almost 2 petabytes of data. If you work out that the speed of processing increases by Moore's law, there's a logarithmic scale, and the storage, the sequencing is still going faster, so we're going to have to work out new ways of processing and analysing that. And to give you an idea of how big it is, if that's a typical movie, which of course I know you don't download, and I know it's illegal, but let's assume you were downloading movies. That's the size of a movie. That's a gigabyte. That is a petabyte. So it's a 1,000,000. That's equivalent to 50,000 movie downloads per second. So that's the amount of data that we're pushing through to keep the instruments running for the whole genome sequencing. So it's not the sort of thing that you're going to be able to have every pathology laboratory doing. There'll be some laboratories that specialise, as we do, and there'll be others that will look at much smaller bits. But certainly the era of just doing a couple of genetic tests in the corner back room, that's not going to be around for much longer. These are going to be specialist centres, because otherwise the cost of this will drive it down so much. And even we cannot cope with that amount of data throughput, so we make use of the academic backbone. We have a range of commercial services that provide certain of these components for us. We are terrified, paranoid about the security there. If someone gets in and begins hacking that or meddling with it, so the security is extreme. And we have got a number of different computer infrastructures, each of which is completely separate from the other, and which communicates with the other through their own separate firewalls with zero trust. And we then make sure that even within that, certain parts of the data are potentially relinkable, re-identifiable to the patient identifiers, and they're only seen by a very small number of our staff who are all clinically trained, and we've also signed confidentiality agreements and have code of conduct, and we generally tend to come from that professional background. And the rest of the data is deliberately de-identified, encrypted, obfuscated, so even if you saw it, it would be of no use to you, as just random junk. So a huge amount of effort's gone to design the secure infrastructure. And what we envisage is that each person who's got 3.2 billion nucleotides, of which 3 million are different from the reference, over time, you're going to want to know how common certain things are or how rare in certain groups of people. And so we're going to want to store that in health records, interrogate health records, and because Australia is just 1% of the globe, we're going to want to collaborate and interact with the rest of the world. And there are a number of standards emerging here to be able to exchange data. So how do you do it? I've spent many years in pathology. I've never seen a pathology system that's capable of doing this. I suspect one hasn't been invented yet. By disclosure, I'm now going to show you how we do it within Genome.One. That's just because that's the only place I know that has got the ability to analyse this amount of data. So I'll just show you for a rare disease how we handle the large amount of data. And the issue is that when we get a referral, patients come in with their complaints and their symptoms, and we get referral notes, as I say, fax or handwritten, describing in reasonable terms, if you're lucky to get clinical notes, what's going on. But the trouble is that the textbooks and the references have got the same terms but with different terminology. And you've got to be, and I think as we saw in our morning session, you've got to be able to just recognise an image or recognise a term and appreciate that those two things are actually the same as those two things. Otherwise you won't match up. And so the way in which we go about it, you've got to train the computers and tell them, give them the dictionaries, that the different terms mean the same thing. So there is a language, a human phenotype ontology, which allows a hierarchy, a structured hierarchy of which terms actually mean the same things, and when you refer to one term, it's a subset or a synonym for some other term. And what we have done is built a system that picks up the language that the doctor might use in sending a specimen to you and classifies this in the HPO, the phenotype ontology terms. We've designed it so it's clinical grade, it's secure, it's encrypted, you can search through the encrypted fields, and the way it is that you just either type in online when you make your request, or if it receives it handwritten, we transcribe everything and we just type it in plain English. And then natural language processing goes through and parses that and pulls out terms it thinks are relevant and maps those onto the human phenotype ontology. So you type in plain English and the computer is in the background working out what are the standard terms. So 3 patients using different descriptions of the same disease will all map to the same disease. It's able to accept images as well as words. Once it's got that, it asks you a few key questions. Look, that term, there's 2 or 3. Can you give me a bit more detail? If you can, there's 3 or 4 variants of that. Just tell me which of these is present in the patient. Once you've got that, you've come up with your phenotypic, phenomics, and then you probability different diagnoses, and then on that you map all the possible genetic causes that could cause that condition. This is all done automatically. And then from that you work out which of the genes could correspond to which of the phenotypic descriptions, and go back to the requesting doctor and say, look, I think I've got to come up with the diagnosis. Does the patient, when you say tall, how tall is tall? When you say they've got long fingers, do they actually have webs in between the fingers? Because you didn't mention that. But now we know that's an important differentiator in the diagnosis. And then from that spits out the genes, links into the journal articles that will give you the evidence to know that's the cause of the gene. Oops. And it produces a report, which we email out, not fax. Just show an example here. Here's an actual patient. I've changed the name. Someone's asked polycystic kidney disease. We've typed in the clinical notes. It's referred to saying there's only 9 genes that fit that. Would you like me to look for the genes? Yes, please go ahead with the 9 genes. We've found a pathogenic variant in the PKD1 gene, which is the most common gene. Here's a description of it. There's the genetic terminology, and there's a synoptic report if you'd like to have it, and it's in a form that it potentially is interoperable with another system when those other systems are built. So we see in our mind's eye that the patient's still going to see their doctor, still have their phenotype, to which we can add the genotype now. They're going to produce the report, have the diagnosis. You'll still have the differential diagnosis. Medicine won't change. But what we'll be able to do is have in the background the omics, the genomics operating, which will be able to make the diagnosis. So the medicine goes on, and we can handle through the background all the growing amount of genetic information, and then simply by interrogating the plain English in the clinical notes or which is in the medical record, be able to suggest which patients will benefit from genomic testing, and for those patients be able to make the diagnosis with a combination, a partnership between the computer and the scientist and pathologist. And afterwards we go through, we give a summary, standard pathology report, we classify them, we give recommendations and suggestions. For some conditions family testing is appropriate and for others not so. Where will it happen? Well, you heard Anthony's description about cancer. We're focusing on the rare and inherited diseases and on health management. Unlike the direct-to-consumer, we're still going out to people who don't have the condition, but we see this as being part of the shift towards preventive medicine. So we're trying to work out which genes, there's an evidence base which will be predictive for those who are going to get various conditions. And we offer now, you may again have seen described in the media last month, the first of the so-called preventive health genomics or preventive health management offered as part of a standard medical consultation, to which we can add a genomic layer as well. Short term, we're only able to do Mendelian conditions, the rare, the classic genetics. There's not enough knowledge as yet for the multifactorial, the common thing, the overweight, the obesity, the diabetes, the blood pressure. It's coming, but it's not there yet. And there's applications from preconception right through life, even through to the molecular autopsy, for those that die of unexpected causes or at unusual ages, to be able to do genetic testing on them to help the relatives who may as yet not have that condition but may be at risk of it. So it takes a village to drive this. Here's some of the 60 people who are there who've developed this and brought it to fruition. That's me up there. It's, for me, as perhaps in the latter part of my career, I'm just delighted that I'm still alive to be able to see part of this. I dreamt about this when I was a medical student. I was told during my medical school lectures it was not possible to sequence the human genome, and even if you could, you couldn't store it. And even if you could, you couldn't interrogate. It just shows you how short-sighted my teachers were. Now here we're doing it roughly every 30 minutes. It's just become... And similarly, our predictions about the future, that we think it's going to become part of routine medicine. People will say in 5 or 10 years, we think it's going to happen a lot faster.